Well, we've been talking about DNA and some biotechnology and how DNA, discovery of DNA by Watson and Crick, that really changed and revolutionized not only what we know about our traits, but also like how to find new medicines for different genetic diseases. All of that would be that, and trying to create new proteins, like for people who don't have insulin in their bodies, for diabetics, they can make new insulin for them. All of this has been done through the use of knowing about genes and DNA and kind of the technology behind how we've discovered those. So we're going to start on this little PowerPoint, and this is a very short one. And so we're going to talk today about what's called gene therapy, which is kind of like what you think of like physical therapy when you hurt your leg or your arm, except that this is therapy to fix genes that might have something wrong with them. And the things that are the proteins that cut DNA into pieces so that you can remove parts that you want and add parts, or remove parts that you don't want and add parts that you do want, those are called restriction enzymes, enzymes that cut DNA in certain places. And you probably know this already, and Watson and Crick kind of knew this, but Darwin even had an idea of this, and that's the term selective breeding, which is another word for artificial selection, where we can actually choose the traits. And we do that with dogs. Maybe people don't realize it, but dogs are all the same species. Horses, too, they're different species of horses, but they look a lot alike. They're in the same group. And we've been selectively breeding, which means we choose what traits we want, and we make dogs or other animals like cats accordingly. So we want the smaller dogs, we breed the smaller ones together. And even Charles Darwin, kind of who we're going to talk about, kind of knew a little bit about this. It's called artificial selection. You're selecting the different traits you want. We've been doing that for years. Dogs too. Dogs are all in the same species, I think I said, and cats too. But now, of course, with the discovery of DNA and with the idea of gene therapy, things are changing rapidly. So you know this. This is called the central dogma theory, and it says that we have DNA that's in the spiral, staircase helix, double helix. We have a process called transcription that turns the DNA into a single strand of what we call messenger RNA, and then that messenger RNA is translated into a protein. And if it's translated incorrectly, or the code for the DNA is wrong, then you get the wrong protein, or you don't get a protein at all. This is called the central dogma theory. DNA turns into RNA, which becomes turned into protein at the ribosomes. So what happens when things go a little crazy? I'm going to let you go to that link later on your own. I'm not going to go through it on the PowerPoint, but there's a link here. I'll leave it up for you for a second. But what happens really is proteins don't end up being made the way they're supposed to. And if there's even one letter of DNA that's not correct, then the protein ends up not working right and won't fold the way it's supposed to fold. And when that happens, it doesn't do its job. So if it's a protein, for example, that's supposed to help you break up your food, then that protein won't get made and you won't break up your food as easily. So what happens in gene therapy is really pretty simple. You take a gene out of a chromosome by cutting it with what we call a restriction enzyme, which we're going to talk about in class, and then you put in a new gene, like perhaps a gene for a protein that you didn't have before, and you insert it in, and then you put it back into your body. So this right here is actually what we call a vector, and it's a thing that is a piece of DNA that we cut, and in this black part right here, that's actually the gene that we want. So when you put it in with a bunch of chemicals, and there's a process that we're actually going to do for this, then you actually can then use that to make copies and copies and copies of that protein that you wanted. So it's very, very, very a very, very helpful thing in gene therapy to take a gene and replace it with a normal, working gene. And then I thought this was really interesting. This is just a picture showing you how many genes there are that work for certain organs in the body. For example, the brain relies on 3,195 different genes on different chromosomes of the 46 chromosomes. And obviously, even if one of those genes goes wrong, there could be some problems in the brain. Same thing with those other parts that you see there. And as I mentioned, restriction enzymes are kind of scissors They're chemicals that cut DNA at specific sequences of letters. They're very precise, and they can be used for pretty much any certain sequence. There's a certain enzyme that will break it. So you could find pretty much any gene that's faulty, and you could find an enzyme that will cut around that gene and cut it out, so you can put a more working gene in. And this is an example of how they might do that. We're going to do this in class, but here's a little section where we have a DNA that will cut an enzyme that will come right here and cut the DNA into a kind of two pieces there. So you can actually merge these two non-similar DNAs together if you have the right enzymes, and you can then glue them back together here. So if you see here, the enzyme cut right here, and this comes over here and joins in with the TTAA here. And that's called restriction enzymes, enzymes that cut DNA at certain sequences so that you can then put pieces of DNA together and glue them together. Now this we're going to do a lab on. This is called transformation. This is where you take a bacteria. Bacteria all have a certain circular piece of DNA called a plasmid in them. And what you do is you go and put an enzyme in there and you cut the DNA plasmid. You insert a gene that you might want. And that's the pink thing here. You take that, let's say, from a human cell, the gene that you want. You put it into the bacteria cell, so now it's part of that. And then the bacteria cells multiply like every 30 minutes. So within days or weeks, you could make millions and millions of copies of a particular gene. Some, for example, might be used to help at fake snow places. When if you go skiing, sometimes they make their own snow. That's a protein that they need to do that. That's added to their machines. Or how about a protein that dissolves blood clots in your body so that you don't have a heart attack? Something clogging up your arteries. Or you can use it to make a protein that would help clean up oil or waste products if there was a waste spill in the ocean or something. And lastly but not least, if you know that plants are being killed by a certain animal or certain a pest, some kind of insect, you can put a gene on there so they can resist that pest. So there's actually a very good therapy for this, where you take a gene out of the human sequence, insert it into what we call a plasmid, a circular piece of DNA, and then put it back into the bacteria and let it multiply. And if you look there, every copy has a copy of that gene in it now. And that's the end of this PowerPoint, and we're going to do a couple labs where we are going to explore these concepts. Thanks, guys.